All right, well, I'd like to welcome everyone to today's edition of the Polyploidy Webinar. Today we have a couple of great talks here on studying genome evolution in polyploid species. And up first is Dr. Sylvia Kinosian. Sylvia's interest in plants started back when she was an undergraduate studying forestry at the University of Vermont. She grew up hiking and canoeing in the Adirondack Mountains of New York State, and initially wanted to pursue a career in land management. However, she became more excited about plant identification, ecology, and ultimately how processes of evolution shape biodiversity. She completed her PhD at Utah State University studying fern systematics and population genetics with Paul Wolfe. And she's currently a postdoctoral fellow here in my lab here at the University of Arizona, working to better understand the evolution and structure of fern genomes. And she's going to tell us today about how genomics is modifying our concepts of evolution in homosporous pteridophytes. With that, I'll let Sylvia take it away.
Thanks, Mike. Yeah, and thank you everybody for coming today. I'll just go ahead and get started. So a brief overview of my talk on is going to be looking at first off how different reproductive modes in vascular plants. So I'll go over the different homosporous versus heterosporous reproductive modes. And then I'm going to talk about some historical perspectives on polyploidy in ferns. And how we got to our current understanding of genome structure and evolution in ferns, but also in angiosperms as well. And then I'm going to talk about some of the work I've been doing in the Barker lab in collaboration with some other members of the lab to understand the unique diploidization patterns in homosporous plants and ferns, specifically using comparative genomics. So first, a brief review on different reproductive modes in land plants. So here's a cartoon phylogeny of land plants, and the purple stars denote independent evolutions of heterospory. So most land plants are homosporous, meaning they produce one type of spore, and heterosporous plants produce two different types of spores. So we have three lineages extant today. So in the lycophytes, we have the genera Selaginella and Isoetes. Of course, we have the seed plants, so gymnosperms and angiosperms. And then within ferns, we have the water ferns or the Sylvaneales. So these are all of our extant heterosporous lineages. And so these plants produce two different types of spores. So they have a megaspore and a microspore. The megaspore develops into the megagametophyte or embryo sac in seed plants, which produces the egg. And then the microspore develops into the microgametophyte or pollen, which produces the sperm. And so this facilitates outcrossing and has evolved multiple times throughout the history of tracheophytes. In comparison, we have the homosporous lineages, and these plants are actually the ancestral condition of all plants. So we have the bryophytes, which are the earliest diverging land plants, but algae and such are also homosporous. And then we also have the Lycopodiales within the lycophytes. And then most ferns are also homosporous. So lineages from the Equisetaales or the horsetails all the way up to the tree ferns, the Cyathiales, and then also the model fern Ceratopterus. So these lineages only produce one type of spore, and that can develop either into a male gametophyte, which produces sperm, or a female gametophyte, which produces eggs or sperm. And so this has some interesting, there's just the one type of spore. And what's really interesting with these two different life history strategies is that they are accompanied by really unique transitions in genome traits. So in all three extant heterosporous lineages, we see low base chromosome numbers and small genomes. So think the small dynamic genomes of angiosperms. The heterosporous lycophytes and ferns also share those genome traits. And then all homosporous plants, so I guess I'm talking about all vascular homosporous plants, have high base chromosome numbers and large genomes. And just a note here, I will not really be talking about the bryophytes today. So those are homosporous plants with small genomes and low chromosome numbers. And I also won't touch on too much the gymnosperms, which have large genomes and small chromosome numbers. I'll cover those a little bit and would be happy to answer any questions about those at the end of the talk. So this disparity in genome structure associated with reproductive mode is something that's really interested me. And my main question that my research has been following over the past couple of years is what is causing these disparate patterns in genome structure across land plants? So I'm going to talk to you about some historical perspectives on polyploidy and genome structure in ferns, as this has been a topic that's been studied for quite a while now. So all the way, this began with work on cytology in plants at the beginning of the 20th century, as well as with Irene Manton in the 1950s, looking at the cytology of ferns and lycophytes. But I believe it was Kwiatkowski and Baker in 1966 that really stated this pattern in a really clear way, and that what they notice here, and you'll see in the figure on the right here, is that about 90% of heterosporous ferns have a base chromosome number less than 28, and 96% of homosporous ferns have a base chromosome number greater than 28. And so you have this really striking divide in the low chromosome numbers of the heterosporous ferns, high chromosome numbers of the heterosporous or the homosporous ferns. And so this led Kwiatkowski to hypothesize that ferns self-fertilize within gametophytes, and so they can have this really extreme form of intragametic selfing, where a sperm and an egg from the same gametophyte fertilize to create an offspring that is homozygous at every locus. And so ferns, just homosporous plants, were polyploid in order to accommodate this high genetic load due to this extreme form of selfing. And so this is through homologous heterozygosity. And so the subgenomes within these polyploid plants were able to compensate for this intragametophytic selfing and being homozygous at every locus. And so you can have shared genetic information through those subgenomes. And so this hypothesis was tested a lot and strongly and supported throughout the middle of the 20th century. And then this is just a reminder of that homosporous life cycle where you can get the haploid spore germinating into a hermaphroditic gametophyte, which can self-fertilize to have this extreme self-fertilization. Anyway, so we're going to jump forward about 20 years to when people started using electrophoresis to understand genotypes in plants. And this work made people start to think that ferns actually have diploid inheritance rather than being polyploid and having this sort of multisomic inheritance to increase heterozygosity and genetic diversity. And so this was through electrophoresis, where you can look at the genotypes of certain enzymes. And in most ferns, they were seeing only 2 enzymes present, which indicates diploidy, rather than multiple different bands of enzymes or alleles present for known polyploid angiosperms. And so this led scientists to think that ferns are indeed diploid and outcrossing with high chromosome numbers. So this was against that original hypothesis that ferns were polyploid and inbreeding as a way to explain their very high chromosome numbers. And so this leads me to the origin of the title for my talk today, which is from this great paper by Christopher Hoffler in 1987 called Electrophoresis is Modifying Our Concepts of Evolution in Homosporous Pteridophytes. So in this paper, Hoffler describes a lot of work that had been done over the past few years from multiple authors using electrophoresis to really understand that ferns are indeed genetically diploid and reproduce via outcrossing. And Hoffler, in that paper, proposed a mechanism for the increasing numbers of chromosomes, or the very high chromosome numbers of homosporous plants. And so he suggested that homosporous plants go through rounds of polyploidization, and they go through cytological diploidization, which I'll talk about a little bit more in a second, essentially a return to diploid inheritance. And then they go through gene silencing, but not extensive gene loss. And so that means that the genetic material is not lost, chromosomes are not downsized. And so now you're left with a diploid organism with a high chromosome number. And then they'll go through another round of polyploidy with the same pattern, leading to ever increasing numbers of chromosomes, despite a return to diploid inheritance. And so this is just a schematic to understand the spectrum of diploidization. And then there's two main aspects of this. So we have cytological diploidization, as I mentioned before, which is a return to diploid inheritance. So you just have two chromosomes pairing at meiosis rather than multisomic inheritance with multiple chromosomes pairing, as well as some physical downsizing and loss of genetic material through genic diploidization and fractionation. And so the genic diploidization and fractionation can be either through gene loss or simply gene silencing. And so what Hoffler was proposing was that ferns were going through the cytological diploidization part, but they were not losing genes. They were rather silencing genes through that gene fractionation process. So I'm going to jump ahead a little bit more, much more recently, when we first started to get transcriptomes and genomes to understand how plant genomes change over time. And a hypothesis that was proposed was that perhaps ferns have more rounds of whole gene duplications than angiosperms, which may explain their high chromosome numbers through this sort of upward spiral of chromosome numbers over time. But through this work, so this is a figure from 1KP, and these numbers have also been corroborated by work in our lab, most recently by Michael McKibben and Jeff Finch, to show that angiosperms have about 3.5 whole gene duplications per lineage through time, and then ferns have about 2.8. And so, if anything, ferns have fewer whole gene duplications than angiosperms, despite having really high chromosome numbers. So just to kind of recap some things that we know about diploidization in land plants is that, at least in angiosperms, they have small genomes, low base chromosome numbers, a few whole genome duplications per lineage, but really fast cytological and genic diploidization. And this has also been proven by empirical studies looking at recent polyploids and how quickly they can downsize in angiosperm lineages. Comparatively, so homosporous pteridophytes have very large genomes, high base chromosome numbers. They also have gone through several rounds of whole genome duplication in their evolutionary history, perhaps not as many as angiosperms. And we're still kind of unsure as to how diploidization occurs. We have this nice hypothesis that it may be through pseudogenation, but I was really interested in looking at this with some comparative genomics to understand what's actually going on here. So now I'm going to talk to you about the work I've done over the past couple of years in the Barker lab. So I had two main questions. And so the first was, how do pteridophyte chromosome numbers change over time? And then also, how does that fractionation process happen in pteridophytes? Are we actually getting gene loss? Are we getting gene silencing? How are those genomes changing following a polyploidization event? So first, I'm going to talk to you about some work I've done with some former members of the Barker lab using the program ChromeEvol to model variation in chromosome number over time. So this figure is going to show you those numbers. And so on the y-axis, you'll see ancestral chromosome number. And then on the x-axis, you will see that plotted as the distance from root to internal node. So I have a cartoon phylogeny on this figure just to show you and sort of conceptualize how these numbers are represented. So from left to right, we're moving sort of through evolutionary time from the root of the phylogeny or the most earlier diverging lineages all the way through to extant species in modern day. So looking at how chromosome number may have changed over time based on the phylogeny. So first, here are the angiosperms. They have a median value estimated of about n equals 10, which is supported by empirical evidence over time from other studies. And we do see a range of chromosome number variation over time. So but all sort of centering around this median value of 10. And then we have gymnosperms, which I mentioned before have low chromosome numbers, but they don't have very many whole genome duplication events, so their genomes don't change a whole lot over time, at least in the sense that we're talking about with diploidization. They have a median value of n equals 11 and very little variation over time, which is supported by empirical evidence that we have. And then finally, we have a really interesting trend for the ferns. So we see median value of n equals 33 for chromosome number, which again, supported by empirical evidence. But we see this increasing trend of chromosome numbers towards the tips of the phylogeny. And so this model is really supporting that upward spiral hypothesis of through multiple rounds of whole genome duplication, of which there's plenty within the history of homosporous burns. We see this increasing trend of chromosome number, which is really exciting to see. So I guess just summarize that we are seeing this increasing chromosome number through time based on these chrome eval modeling. So moving on to how does fractionation happen in pteridophytes? So we want to understand if gene silencing or gene loss is a major factor in how genomes downsize over time following polyploidization. So here I'm going to show you some syntenic block distributions. You may be familiar with these, but it's essentially a histogram. So on the y-axis, we have the frequency of the block size, and then on the x-axis, we have the actual syntenic block length. So a syntenic block is just genes that are longer syntenic blocks indicate that the genome has undergone less restructuring and has broken up less following a polyploidization event. And shorter syntenic blocks indicate that it has broken up more and been has changed and downsized over time following polyploidy. So just for reference, I'll show you a syntenic block distribution for angiosperms. And so these are actually data from some great work by Michael McKibben. And so angiosperms have a lot of really small syntenic blocks, which makes sense because we know that they downsize really quickly over time. So now I'm going to show you the same for heterosporous pteridophytes. And perhaps unsurprisingly, we're seeing a really similar pattern here of a lot of small syntenic blocks, indicating that these genomes have downsized rather rapidly and broken up quite a bit since their last polyploidization event. But yeah, this makes sense. We know that heterosporous plants all typically have really similar genome structures. So this was good to see. What was really exciting is that we see homosporous pteridophytes actually show a really similar pattern of small syntenic blocks, which is interesting because their genomes are rather large. And so this is indicating that we might actually be having some gene silencing aspects of restructuring the genome rather than just gene loss, just because we know that their genomes haven't really downsized all that much following polyploidization. And then just for comparison, this is all pteridophytes. So this is about 12 published genomes, roughly equal of heterosporous and homosporous plants. And so we're seeing this really similar pattern to angiosperms, despite the fact that we know that their diploidization is probably fundamentally pretty different. Some further evidence for this is from the Ceratopterus genome. So Ceratopterus is the model fern, and its genome came out a couple years ago. And this is a similar syntenic block distribution for Ceratopterus compared to angiosperms. And so we see that Ceratopterus itself, it is a homosporous plant, but it looks like it has a ton of small syntenic blocks, indicating relatively fast diploidization and downsizing. And then I also want to draw your attention to this inset plot. So essentially what this plot is showing is a metric for diploidization rate. So the closer you are to the lower right-hand corner, the faster the diploidization rate is. And so what you're seeing, all of these blue dots are angiosperms, and then the orange, lighter blue, and green dots are all ferns. So we have Ceratopterus in orange, which is a homosporous fern, and then we have Sylvinia and Azolla in green and blue, which are heterosporous ferns. So what's really interesting is that we have these two different reproductive modes of ferns, but they're all downsizing really quickly. Which is really interesting. We might expect that for heterosporous plants, but not for Ceratopterus, which is homosporous. But that it may be an outlier, but I think we will see more in the future. Anyway, so what we're showing here is that homosporous and heterosporous ferns are all downsizing pretty quickly, but perhaps via different mechanisms. Further evidence for those different mechanisms comes from collinearity plots. So this is genes that are in the same order between two different genomes. And so this is two homosporous lycophyte genomes, so Diphasiastrum and Hupersia. And this paper came out this year, and it's a really beautifully shows that the gene order between these two species is remarkably conserved, given that they have about 350 million years of evolutionary time between them. So we find a really similar pattern in homosporous ferns. So this is Ceratopterus, again, that model species. And then we also have Alsophila, which is a tree fern, and it actually has incredible intragenomic synteny. So between its two subgenomes, it's incredibly conserved. And despite Ceratopterus having pretty rapid genome evolution, it also has a lot of conserved collinearity between Alsophila, despite these two species having about 250 million years of divergence between the two of them. So how does fractionation happen in pteridophytes? I don't know if we have exactly the answer now, but we do know that fractionation happens relatively rapidly, which is interesting to see despite the fact that their genomes are so large. And we have this really striking preservation of gene order or collinearity in between genomes. So all together, we see increasing numbers of chromosome numbers over time, and we see perhaps gene silencing rather than gene deletion as a mechanism of diploidization in homosporous pteridophytes. So this is some really cool comparative genomic support for the upward spiral hypothesis proposed by Hoffler in 1987, based on electrophoresis evidence. Yes, this has been a really fun project to work on. I did want to give you some caveats. So the genomes that I'm using right now they are perhaps on the opposite ends of the spectrum for diploidization in pteridophytes. We have really fast diploidization in Ceratopterus, Azolla, and Slovenia, and relatively slow diploidization in Cyathea, Huperzia, and Aphasiastrum. And so, of course, this is probably an evolutionary spectrum. We only have about a dozen pteridophyte genomes now, so about roughly the same representation of heterosporous versus homosporous pteridophytes. So hopefully with more genomes, and there are several on the way, which is great. Hopefully with some more of this pattern of diploidization rate and mechanism will become a little bit more clear with time. And then looking towards the future, I'd really love to look at what mechanisms are actually causing these different patterns of diploidization. So perhaps looking at things like the dynamics of meiosis between homosporous versus heterosporous plants. And then looking at how life history traits, so something like growth habit, might affect genome evolution. It's known that these can affect molecular evolution rate, so perhaps they're doing something with genome evolution as well. And then finally include bryophytes. I mentioned in the beginning that I wasn't really going to talk about them, and it's because we don't know a whole lot about them with their diploidization right now. We do know that they have a wide range of whole genome duplications between the lineages of bryophytes, but overall, a relatively few whole gene duplications in their histories, and they have very different selection pressures on their genomes than vascular plants. So hopefully in the near future, we'll understand a little bit more about them and be able to include them in these comparative analyses. So with that, I would just like to thank my lab and funding, and I will be happy to take any questions.
All right, well, thank you, Sylvia, for a great talk. Yeah, if you have any questions, feel free to raise your hand or put them in the chat, and we'll go from there. Great. Well, if there are no other questions for Sylvia this time, feel free to keep those in mind as we come back at the end. Will be a chance to ask some other questions if you think of them. And with that, maybe we'll switch over here to Tamsen. All right. Thank you, Sylvia.
All right.
All right. Let Tamsen get her slides shared here.
Okay, hang on a sec.
All right. There we go. All right. Well, I'll go ahead and looks like we're getting that up and running. I'll get you introduced here.
Thanks.
All right. All right, well, I'd like to introduce our next speaker then, is Tamsen Dunn. Tamsen's a 6-year PhD candidate in the evolutionary biology joint doctoral program at San Diego State and UC Riverside. She holds a master's degree in applied mathematics from UCSD. And after working for 10 years as a bioinformatics scientist in industry and doing habitat restoration during her vacations, Tamsen felt it was time to combine her research with her lifelong passion for plants and conservation. In the Seth Raman lab at San Diego State, Tamsen is developing new computational methods for the study of polyploidy and evolution. She enjoys rock climbing and camping with her favorite research assistants, her twin daughters. And with that, I'm going to let Tamsen take it away here and tell us about her work on deciphering the polyploid KS histogram with forward time simulations.
All right, thank you so much for that nice introduction. I'm going to stop sharing and restart just one more time because it's looking a little funny from my end.
Yeah, it's the whole screen
Okay.
right now. There you go. These are the fun parts of us running Linux Zoom. All right. Hopefully you can pick that. Well, I find that it usually works pretty well to pick the whole window and then, but anyway.
Can you see? No.
Nope.
All right, I'm going to share again.
It was working when we did the practice run earlier. So, that looks like it'll work.
All right, we'll go with this. Can you guys see my screen okay now? Acceptably?
Looks good. And I think
All
if you
right.
make the presentation mode there, it should work. All right.
Yeah, sorry, Zoom updated. I knew it was going to do something funny this morning. All right, thank you so much for everybody who's online today. I really appreciate your time and listening to my talk. So I'm going to tell you about deciphering the polyploid KS histogram with forward time simulations. And this actual paper was just accepted yesterday to MBE, so I'm pretty excited about that. As I'm sure this whole audience knows, whole genome duplication is replete across the tree of life, and we see it a whole lot in plants. Allopolyploids are polyploids that are derived from two different species, and those genomes have come together in the polyploid. And then autopolyploids are derived from whole genome duplication that originates within the same species. And one of the major impacts of this difference in mode of origin is that only in allopolyploids do orthologs, which are genes that have been separated by speciation for some amount of time, later become paralogs. So when we talk about KS, and Sylvia mentioned that a little bit today too, when we talk about KS, we're talking about the average number of synonymous substitutions that have been accumulated between those pairs of genes over time. They also accumulate non-synonymous mutations, but we're going to be talking about synonymous because they are kind of excused from selection for the most part. So we can kind of use the accumulation of KS as a proxy for time. So even though these genes do kind of take a random walk away from each other, random walks are on average predictable. So with a little bit of hand waving, we can see that the accumulated KS lets us know how long these genes have been separated for in time. So the inspiration for my project actually came from a Lee and Barker paper from 2020, where Michael came to San Diego State and showed us these amazing KS histograms across the plant kingdom. And you can see they make this great kind of variety of shapes right here. And that's because the KS histogram can be impacted by many parameters. Historically, we've used peaks in these KS histograms to infer that whole genome duplication has occurred, but there's other parameters that we can theorize might also impact the shape of these histograms. So while that's a problem, if we're trying to use these histograms in isolation to figure out when whole genome duplication occurred, maybe we can turn the problem into a solution because, oh no, these histograms are messy, but why are they messy, right? What can the complications and the information in these histograms actually tell us about the history of these lineages. So with a very simple model, you can think of what shapes the two main processes that shape these KS histograms. So first off, there's these small scale duplications, right? So genes are constantly being born and constantly shed, and they're kind of erupting in isolation. And that follows an exponential curve, an exponential decay, rather. So you can imagine the genome as just being like a bucket of paint with a hole in it to represent gene shedding. So there's a faucet here, and genes are continuously being born. So they're being poured into the bucket. And they're also being continuously purged because there's, a deleterious effect or a cost to having DNA you don't need. So there's a bucket of paint, and then the amount of time the paint actually sits in the bucket is how long those heterologous genes have to accumulate distances, genetic difference between them. So that's the accumulation of KS. So if we're just talking about small scale duplications, we just have genes coming in and genes dying. But when whole genome duplication has occurred, what's happened is sometime in the past, imagine somebody's just dumped a big bucket of black paint in this bucket, 3 days ago. Well, you'll still see some remnants of black paint, but it's been leaking out all this time. And the black paint is going to have a characteristically different age than the red paint because it all came into being in history 3 days ago, right? So that's why we've got a big peak here in the KS histogram from some past whole genome duplication. So when we're talking about using these KS histograms to infer whole genome duplication, that's what we're talking about. That's what we're looking for. But there's some problems with this. So here's our KS histogram, and I just explained the small scale duplication peak and the whole genome related peak right here in the paralogs. But when we're trying to use this to actually time when whole genome duplication occurred, there's a problem because sometimes the time at which we start the clock on accumulating these differences between the paralogs is not the time of whole genome duplication. And some people have found that to be, completely obvious, and it's in the literature, since there's a paper by Doyle from 2010, and I think Mike said there was a paper from Gott, maybe even 10 years before that, that said, yes, you're going to have problems here because when you time whole genome duplications by KS, what you're picking up is when the divergence time between when these genes started, which for allopolyploids is actually when the parents diverged. So your error in your estimation from using the KS distribution to estimate your time of whole genome duplication, you're going to have an error that's going to scale with the difference in time between your whole genome duplication time and your divergence time. And for plants whose parents have been diverged for 10 to 20,000 years, it's about maybe 5 to 10 million years before they come back together, that can be a problem. And we have evidence of plants hybridizing even after 50 million years. So, you want to be really careful. And that's, we obviously don't just use KS in isolation to time whole genome duplication events anymore. There's a lot of other means we can use, like synteny, or as Mike McKibben talked about last time, we can use extra information from phylogenetic trees to help pinpoint those whole genome duplications. But in any case, the big problem really arises from what I alluded to in the beginning, the difference between autopolyploids and allopolyploids. So I think canonically people were just thinking of polyploids as autopolyploids, where there was an ancestral lineage and, it's evolving through time, and suddenly its genome duplicates. And around that time is when the paralogs are able to start accumulating KS. So you see that KS peak maybe about when whole genome duplication occurred. Although there's very compelling arguments to state that maybe it actually happens even further after whole genome duplication for autopolyploids. Meanwhile, for allopolyploids, here's your ancestral species. Here's two parental species diverging, and then they come back together to be the polyploid at some potentially significantly offset time. So KS accumulates right here at the divergence of the parental species. And so if you're going off this KS peak to infer when whole genome duplication happened, you're going to be wrong. And you're going to be more wrong the more allopolyploid your species is. So yeah, I guess this is essentially what I just talked through. For autopolyploids, divergence between those paralogs is going to happen after whole genome duplication. But for allopolyploids, the divergence is going to happen before whole genome duplication. So what you're getting here is the divergence time, not the whole genome duplication time. So trying to resolve the differences between divergence time and whole genome duplication time led me to think of polyploidy more as a 2D continuum. And when we want to model it and figure out how to recreate those KS histograms, we're going to have to take these factors into account. So we need a way of describing allo and autopolyploids, not just as a 1D continuum, but as at least 2-dimensional continuum where you can take into account the difference between divergence time and whole genome duplication time, and then also the distribution of the ortholog coalescence for either the allopolyploid or the autopolyploid case. So there's kind of two parameters at a minimum that we need to take into account. So here's kind of a cartoon of that. So the delta T here is the difference in time between parental divergence and whole genome duplication. And then the x-axis here is ancestral diversity, because remember we talked about the distribution of that KS peak and the ancestral species when the parents diverged. Well, what's going to give us that distribution of divergences? Well, one theory is you can use the Kingman coalescent, because that tells us within a species, at least if that ancestor was in panmixia, we do have a distribution that we can start with that will actually even tell us something about the diversity of that ancestral species. So here's that 2D continuum, kind of pictorially. You can think of whether the ancestral species had a high or low diversity, which would affect the coalescent time of those original gene duplicates that will go on to become paralogs in the duplicated lineage later on. So my project idea then was to build a simulation based on this 2-dimensional continuum to model KS distributions and see if we can build more realistic distributions. And then given that, if we can control all the parameters and then build a KS distribution, can we then give an empirical KS distribution, go back and infer the original parameters, and then to provide an accuracy assessment? So I modeled the onologs and the small scale duplication, so the black paint and the red paint basically. And these topologies, you can essentially model kind of using the same framework. All you need to do is put a divergence time in and then allow some distribution model for the onologs. So in my model, I allow the user quite a lot of freedom with what kind of distribution they want there. So given the parameters and the simulation, I'm able to generate a large range of KS histograms that have a pretty characteristic shape. The simulation has 8 kind of modules to it. So we generate a species tree, and then within that species tree, we fit gene trees. They go through a gene birth and death model so that every gene that's born has a kind of death date immediately assigned to it based on an exponential model. Then we shed genes depending on how long the polyploid has been a polyploid for, because we don't start the clock on gene shedding until whole genome duplication. And that's important. You don't want to get that confused with the parental diversification time. Right, then we evolve the genes along those gene tree topologies using Evolver. and do a KS calculation, and that's all done by PAML. Then you can generate the histograms. So this is what this looks like. So if I pick kind of 4 points within that 2G continuum, you can see across the bottom here, as we increase ancestral diversity, those KS histograms kind of get a much fatter tail. And what you're looking at here is kind of the effects of stochasticity on the Kingman coalescent of the ancestral population that generated that original ortholog diversity. And then when we look along the y-axis, we can see the difference in divergence time versus whole genome duplication time really affects the amplitude, basically the area under the curve here, which is what you'd expect because the remaining genes are the area under the curve. So more recent whole genome duplications have had less time for gene shedding. So you're going to expect a larger peak here with respect to the small scale duplications, which are up here at the front. And then I wanted to see how these plots fit empirical KS distributions. So I took 3 well-characterized species, coffee, corn, and populace, with a range of whole genome duplication time. So coffee, which is a fairly young polyploid, corn older, and populace, much older, around 65 million years ago, just to see if this model kind of fits the expectations. And so we can see that, yeah, it does. And the curves actually fit pretty well with what we predicted. Lastly, another thing these simulations show is that there's a better way to figure out time of whole genome duplication. So, don't use the peak of the KS histogram, but if you use the area under the curve, if you use the number of gene pairs remaining, then you can get a much better estimate because you only start the clock on gene shedding after whole genome duplication, where the clock on accumulating KS starts at parental divergence. So for allopolyploids, at least under our simulations, it would be much better to go with gene shedding. And then another thing we wanted to use this model for was our results indicated that the kind of fatter the tail is, the more diverse that those ancestral parents were, the more deep in time that coalescent went. So when we looked to see how the shape of those empirical KS distributions from the 1KP data set matched up with our predictions, so we took over 200 whole genome duplication events that have been detected in the 1KP data set and classified them, given our model, to see if they were more or less allopolyploid, depending on how diverse the parental species were. We saw that about 95% of them seemed to have quite diverse parental species, and that was concordant with Wang et al. From 2019, who saw that it seemed like most plant genomes were likely derived from allopolyploidization. And so this software is up now on bioRxiv. It's just recently accepted at MBE. Sorry, the software is on GitHub. The paper's on bioRxiv. And yeah, it should be an MBE hopefully soon. The assumptions that I used were pretty simplistic. So there's no selection, there's no tandem duplications. So the next step I have is to extend the model. So while this fits for like a single whole genome duplication and a fairly clean, small scale duplications, we have these other more complicated KS histograms. For example, here's one with multiple peaks and these kind of shapes that don't really fit our predictions very well. So what's going on there? So to add multiple whole genome duplications to the simulation I have, that's not too difficult, but I'll be working on that. And then also I want to include the effects from population structure and selection. So that's what I'll be working on in the coming months. So thank you all for your time, for listening to me and your support. And I also wanted to give a shout out to Brittany Sutherland and George Tiley and Paul Plishak and Zheng Li and Michael Barker and Matt Hahn, and a lot of people whose work and side conversations definitely inspired me in this effort. So yeah, with that, I'm happy to take any questions.
All right, well, thank you for a wonderful talk, Tamsen. Yeah, anybody has any questions, feel free to raise your hand or drop it in the chat. I know there were some other questions that were directed at Sylvia there during the middle of the talk. So feel free to do that. Feel free to either put it in the chat or raise your hand on the box. And I have a real quick question while other folks are thinking of things. How's that? So first of all, I think this is awesome and so fantastic. And to see this modeling. One of the things I was curious about, you're talking about that, in particular, potentially using the gene shedding rate as a way to estimate the ages of these polyploidies might be a little bit of a better approach than trying to place these peaks in time, right? When you model the gene shedding or the gene loss in the SpecKS framework, is it a continual, is it like a single linear rate that you're modeling, or what's the distribution of that model?
Yeah,
that's what I was wondering about. Yeah.
Exactly. So it's just a, single rate, and then we fit an exponential distribution to, how it occurs. So yeah, we don't allow it to vary over time. We could extend it as a feature. And also it's going to be lineage specific too. So I think
that's the thing. Yeah.
Quite a limited utility. So within the framework of our simulation, it fits pretty well. But I would definitely not recommend anybody use this in isolation.
Yeah, that's cool. Yeah, I was curious about that. I think that, there's, it would be fun to think about, well, when we talk about this later too, how to make that a little bit, realistic in terms of the and test, and also test then as a way to test how rates of gene loss change over time following polyploidy. So.
Yeah, absolutely. Maybe as a null hypothesis, it might be a good way to start.
Yeah. Yeah, exactly. Very cool. Any other questions for Tamsen? Or for Sylvia?
Can you hear me?
Yes.
Hey there, great talk. I'm just wondering how the model is going to handle not only the polyploidization, but polyploids, like later after they form the polyploids, they can have reticulation. Is it able to do something for the polyploids and plus the reticulation, the networks?
So we handle a primary reticulation for the allopolyploids, right? The parental divergence and then coming back together. But a secondary reticulation is something that I haven't modeled yet. But I think the framework is pretty good for building that in.
Cool, thanks.
All right, are there any other questions for Tamsen or Sylvia? Okay, well, if not,
I had a question for Sylvia. Sylvia, do you know, is there any adverse fitness consequences for those ferns as they accumulate massive amounts of DNA? Because you mentioned it's protective to some level, but is it kind of, trying to weigh against any adverse consequences?
Yeah, that's a great question. I'm not really sure. I do, obviously having a large genome has some detrimental effects, like your chromosomes can only be so big, or your genome can only be so big because that increases time with things like meiosis and mitosis. But I'm not really sure what's going on with ferns because they do seem to have these massive genomes with relatively few consequences, perhaps consequences other than just having a lower overall genetic diversity than angiosperm. So something is going on there, but we have within homosporous ferns, you have both the largest chromosome number, so that's an Ophioglossum species or the adder's tongue ferns, and then also the largest genome size, so that's Deciphering. I don't know, they're a bunch of little weirdos, and I wish I had a better answer for you, but I think that will definitely be something to look into in the near future.
Thanks. Yeah, that's super interesting.
All right. Well, with that, I'll just like to wrap it up and thank everyone for coming out today. I appreciate both Sylvia and Tamsen taking time to put together these talks and talk to us. I know that it's a busy week with lots of things happening, and proposals and all sorts of things going on that folks are working on. So I appreciate both of them for working on this, as well as everyone else for showing up. I'd also like to highlight our next, continuing with our first Wednesday of the month schedule here for the Polyploid webinar. Our next talk will be on December 4th, and it'll feature Doug Soltis and Trevor Fasky talking about polyploidy in particular, and I think Doug's going to talk about the Polyploid institutional, the BII project that they have going on, and introduce that to the community. And Trevor's going to talk about variants to similarities in mixed ploidy genomic data causing irregular patterns in the PCA and other clustering analyses. And so please feel free to show up to it again next month. Hope everyone has a great few weeks, and we'll see everyone then. Thank you.